Good afternoon, everybody, and welcome to the seminar series. In Hillary's absence, Hillary just having gone off to Harvard on secondment as of today, it falls to me to introduce Martin. And when I saw the title of his talk, which you've got up on the screen here, What Have Genomes Ever Done for Us, and How Population Genomics is Revolutionizing Our Understanding of Resistance, it took me back a little bit in two respects, I guess. It doesn't seem 5 minutes since I was sitting in a room with Frank Collins, Nora Bozansky, Fotis Kafartos and several others talking about whether or not we should try and get the Anopheles genome sequenced, who was going to do it, who was going to lead it, and who was going to pay for it. Because to get one genome sequence then was many millions of dollars and a huge amount of work and a big technical problem. And as I'm sure you'll be hearing from Martin now, doing 1,000 mosquito genomes is very cheap, or relatively cheap, relatively straightforward, relatively easy with the technologies we've got now. So a huge shift over that time period. But it also takes me back, specifically introducing Martin, in that when I first arrived here, I was told that there was a young lad who the school had taken on. He'd done his PhD here, I believe, with Harold. And he wasn't here right now. He'd been sent off on secondment to CDC, but was due to come back as a lecturer at the school. And so I think it was about a year before I actually met Martin after I'd been here. But Martin was the young lad who'd been taken on at the school as a lecturer, and now as professor of molecular, I can't remember the full title, but certainly molecular is in there on the entomology. I think it's got something else in there as well. Molecular population entomology, is that right? Too many complicated titles, yes. Very close, but not quite. But I'm sure we're going to have a really entertaining talk. So, Martin, over to you.
Thank you. Okay, so I guess the background to this talk and why I was volunteered was that for the last 6 or 7 years, I've been working with folks at the University of Oxford and Sanger, and many of us in this room have been working on the same project, on a program called the Anopheles gambia 1,000 Genomes Project. And it was just pre-Christmas when we actually finally managed to submit the flagship paper from that work, and it's now on bioRxiv for those who are interested who want to follow up on some of the stuff we're talking about. So it seemed an opportune moment to say, well, what have we and a large number of PhD and postdocs been doing for the last 6 years, and sort of give you an understanding of where the 1,000 Genomes Project is. And I thought it was also quite important to flag that as we develop the new research strategy, or strategy for the school from the 2017 onwards, which has got a very strong emphasis on translational research, try and give you a sense that there is I think, a place for genomics, and there's a real place for the school to lead on some of the sort of hypothesis-driven genomics. I'm not suggesting that we as a school are ever going to rival Sanger, Beijing, the Broad as a sort of a sequencing institute, but I think we have the skills, and by we I mean the people here, but also our network of collaborators, have the skills to actually really define the key questions which we need from an epidemiological or biological perspective. So the somewhat facetious title refers to a charge which is commonly leveraged at genome studies. We've had the genomes essentially of all three major actors within the malaria life cycle for the last 15 years. And people from possibly outside the field always question, well, what have been the major public health advances? And for those of you who don't read the Daily Mail, there have really been relatively few molecular markers which are strongly predictive of disease states, or in this case, other important phenotypes identified through these genome studies. I think the identification of the ginger gene is obviously particularly attractive because it shows it has a pleiotropic effect, and it's also associated with fiery temperature, and apparently Chris Evans' bad behavior as well. So whilst the Daily Mail seems to be convinced that we've identified a lot of genes in general, that's not true. And what's been an emergent property is that we're starting to see that there's lots of genes which are associated with phenotype, and they all have small effects. So we're not seeing these major genes. So that's obviously a trivial example. What have we observed in malaria itself? So this is a study led through Sanger by Dominic Kwiatkowski's group. And this is a genome-wide association study. So it's looking at children in the Gambia who are either classified as having uncomplicated malaria or severe malaria. They are genotyped at 500,000 SNPs using a gene tip. And what we're looking at here, this is known as a Manhattan plot. We'll show you a few of these as we go along. And so we're scanning across the human genomes here in these children. We're looking at individual DNA polymorphisms in each of the chromosomes. So this is chromosome 1, chromosome 2. And what we have on the y-axis is a level of significance. And it's a log to the base 10 or minus log to the base 10 of the p-value. And so on these plots, the highly significant SNPs or polymorphisms are up here, and these are marked in red. So what we're looking for is pile-ups of SNPs which seem strongly associated with one or other of these disease states. So this study, as I say, was looking at severe malaria, any inherent human risk factors for severe malaria. And there's a lot of additional work that was gone on here, so it's been a fantastic study in understanding how to do genome-wide association studies in high diversity African populations and imputation methods. But what about this key question? What were they able to identify that risk factors for disease severity in human populations? And you can see there's a pile-up of hits around here, which seem to be strongly associated with disease severity. Any guesses as to what they may be? What's the common trait associated with disease severity? Okay. It's sickle cell. So sickle cell trait has been known for very, very many years to be associated with malaria risk and infection risk. So these maps are showing sort of endemicity of malaria in sub-Saharan Africa, and overlaid over here we see sickle cell trait frequency, strong association, and it's over 60 years since Allison first demonstrated that patients with sickling cell had far lower infection rates. So the data which came from that GWAS study are fantastic in that they show a confirmation of a known resistance-associated variant, but they're not actually illustrating, at least in that first study, many new underlying traits. And so that's a common criticism I think has been leveled at some of these studies. But I don't think it's fair to critique other people's work unless we sort of shine the same sort of light on our own. And a lot of the work we've been doing is associated with resistance. So to put the genomic work we're doing into a public health context, data from Peter Gething's group has argued that in the last 15 years, we've probably averted, through various public health interventions, approximately 260 million clinical cases of malaria. And what this graph is showing is what's the attributable fraction attributable to ITN use, ACTs, or indoor residual spraying. And you can see that 68% is estimated of the cases which have been averted are attributable to ITN use. So to put that another way, some data from the GPERM document, which estimates at present level of coverage, complete loss of pyrethroids, the insecticide used to treat bed nets. So in essence, a bed net then just becomes a physical barrier, could lead to an increased annual malaria attributable deaths of 120,000. So it's a major problem confronting us, and that's the sort of problem we work on. And what we're particularly interested in is identifying the mechanisms of resistance in mosquito populations, both so that we can develop insecticide resistance diagnostics, which can be used in resistance management strategies, but also through that mechanistic understanding, we can understand how resistance evolves and how cross-resistance is likely to occur. So this is a schematic of how an insecticide might affect an insect. We see these little black bobs are insecticide, which penetrate into this yellow block, which is our cartoon of a insect. A small fraction of the insecticide is excreted, a small fraction is metabolized, but most of it goes on to affect the target sites, and these are mainly neurotoxins, resulting in death of the insect. And what we've been interested in understanding is where resistance occurs along this pathway. And there's some very intractable things like behavioral resistance. We don't know how behavioral modifications have a very good understanding of that. We're starting to see evidence of increased cuticular thickening, which reduces the penetration of insecticide. But resistant insects are commonly showing increased excretion, higher levels of detoxification, and these target site changes in the target of the insecticide. And what we've been able to do using sort of comparative genomic approaches, exploiting the genomes which are, say, published 15 years ago, are identify resistance variants associated with resistance. So we now have a large number of resistance markers for mutations which cause conformational changes in these nerve, either voltage-gated sodium channels or acetylcholinesterase. But really, what we're looking at here is essentially this is the single new marker we've been able to develop in the last 15 years. These markers here were developed just as the genomes are coming out about 15 years ago, and in that 15-year intervening period, this is one of the few validated markers. And that means there's a lot of unexplained variants occurring in these. So we have a very small, explain only sort of maybe 15 to 20% of the variants in this phenotype, this resistance phenotype. And so what we, by we, this is a universal, we of the insecticide resistance group across the school, have been looking at is using transcriptomic approaches to try and understand what's going on in this compartment here of metabolic resistance. And the need for this is because the markers we have, this relatively small number of markers, have not really been widely pushed into public health use. They have they are reliably associated with resistance phenotype, but they don't have a sufficiently strong predictive value to actually be used for replacing simple phenotyping assays. And as a consequence of that, what we've observed for many of our markers, and this is really just a series of our own and other people's researchers' work, documenting allele frequency change over time. Essentially, we've taken longitudinal studies and watched these things accelerate to fixation. So these markers have become essentially useless for predicting interspecific or intra-population variants in resistance. And that's what we really don't want to happen. We want to have a suite of markers which have strong predictive value and which we can use, I say, in resistance management strategies. So digging down into these, understanding the metabolic resistance mechanisms and vectors, using both Janet and Hilary and ourselves, Dave Wheatman, have developed whole genome microarray, or latterly candidate and then sort of latterly whole genome microarray approaches. And what they've come up with is, in some sense, quite a comforting. A relatively small number of, in this case, cytochrome P450s seem to be reproducibly associated or overexpressed in resistant individuals. So we are quite hopeful that we're seeing repetitive overexpression of this small number of loci, so we have potential for genes of big effect for which we could develop resistance diagnostics. We had data from some of our earlier genome-wide scans of resistance that selection imposes can sort of cause a large scar on the genomes of these insects, and really just showing you some data from a comparative study of Anopheles gambiae and Anopheles kalitzii, or in this case, which are labeled M and S form here. These are two sibling species, and what we're looking at here is something analogous to a Manhattan plot. We're scanning across the genome of these insects, and we're looking at sort of an estimate of differentiation. And so this is an interspecific comparison, and what you can see, there's reasonably high levels of differentiation across the genome, but there's a big peak here. And this peak is associated with a resistance-associated variant, which is found in one species, but not the other. However, what we do find is this introgresses, it jumps the species barrier, if you will, into the other species. And so some M-form individuals are wild type, but some now have this KDR, this resistance mechanism. And you can see it leaves this big trace of differentiation in the genome. And this is around 3 megs. So we were confident that we had a relatively small, our transcriptomic studies were showing we had a relatively small number of resistance-associated genes, these principally the cytochrome 6 P450s. And we are confident from our looking at these sort of voltage-gated sodium channel, a known resistance variant, that we were expecting to see a big signature of selection in the genome. So we proceeded with developing a genome-wide association approach, very much like the severe malaria study, but in this case we phenotype mosquitoes for susceptibility to an insecticide, and then we genotype them at around 1,500 SNPs. And these SNPs are in various candidate gene families, the GSTs, the carboxylesterase, and the cytochrome P450s. And what we're trying to find is SNPs which seem to segregate with one or other of our phenotypes. I say very much like the severe malaria study. And so what did these studies find? I guess those of you aware of sort of publication strategies, the fact that we've published this eventually in PLOS One probably indicates we didn't find a lot. And that's true. We were able to replicate an association between SNPs within the voltage-gated sodium channel with resistance to pyrethroids in Ghana and Cameroon, but very little else popped its head above a sort of a reasonable significant threshold. So whilst this is, again, very much like the severe malaria story, a good validation of the approach in that we are coming across knowns, if you will, we're not able to actually discover many new novel associated variants. It hasn't all been doom and gloom for these approaches. In studies in Uganda, we were able to uncover some novel resistance-associated variants. This is a study of pyrethroid resistance in eastern Uganda. And what that has led to is two new resistance-associated variants which are reliably associated with pyrethroid resistance in the region, so both in western Kenya and parts of western and eastern Uganda. So what we're showing here is the two variants, cytochrome, so it's 4J5 and this carboxyl esterase. This is in essence similar to a Manhattan plot, and what we're seeing here is anything which above this dotted line is a population whereby we can replicate that association with pyrethroid resistance. So we have, in the last 15 years, managed to now, we've probably got a suite of 6 new markers which explain, so the 3 markers here probably explain 20 to 30% of the variants in a pyrethroid resistance phenotype, but there still are, say, an appreciable fraction which is unexplained. And hopefully, I'll talk now about the 1,000 Genomes Project and how it's going to take us, I think, remarkably quickly towards trying to complete the levels, to try and explain more of that variance, that genetically determined heritable variance in phenotype. So the 1,000 Genomes Project is a large consortium project led by ourselves and Sanger and University of Oxford, but a large, I'll say, a consortium of various people. These are the major drivers of the analyses I'll be talking today. And the idea was that we would collect Anopheles gambiae, the principal malaria vector in sub-Saharan Africa, from across its range, or as far as possible from across its range, characterize natural genetic variation, develop web-based software for actually interrogating that variation, and try and start to understand its population structure and also medically important traits. And so we obtained samples from across sub-Saharan Africa, and this is the distribution of those. So each of the red spot is a population sample which has been contributed to the 1,000 Genomes Project. Some of these have associated sort of phenotypes in that we know sort of these are populations which have high levels of insecticide resistance. We know, for example, these populations from eastern Kenya and eastern Uganda are separated by the Rift Valley. So we can make some a priori, some predictions that we're likely to see marked discontinuities in population structure across this. But in general, what we were trying to do is sample as widely as we could across the range. And in the first phase of this data, the data which are released here, we're talking about 765 wild-caught individuals, so approximately 100 from each of 8 locations. These were sequenced down at the Sanger on HiSeq to an average coverage or notional average coverage of 30x. And in general, we don't have individual phenotypes for these mosquitoes, but we do have some sort of ideas about their population phenotypes, if you will. And when I gave Diederik the title, I said I'd be talking pretty much strictly about resistance, but I thought it might be worth, well, I hoped it would be worth to talk a little bit more general about some of the broader implications for this work. So I'll talk about 3 different studies which are conducted with these data. One which looks at characterizing genome variation and how this might impact some of the new genome editing technologies which are being proposed as alternative control strategies. Whether we can use these data to monitor a suppression of vector populations, and also talking about sort of the evolution of insecticide resistance as well. So we took 765 individuals. we perform really quite stringent accessibility criteria, and using those, we can estimate that we're characterizing about 91% of the exome of these species. And within that 91% of the exome, approximately 63% of the genome, we've discovered about 52 million SNPs. To put that in context, that is shown down here. And what we're showing here is variant allele density. So we're scanning across the genomes. There are regions of the genomes around the centromere, which is what this cartoon here is showing, which have lower diversity. But in general, what you can see is, apart from outside these centromeric regions, what apparently are quite high rates of diversity. And what that diversity is a variant allele density of 0.5. So it means that we see a polymorphism every other base pair. So in this relatively small subsection population, 765 mosquitoes, if we scan across that subset of genomes, we will find a polymorphism every other base. And so perhaps another way of putting that is that this 52 million SNPs is an order of magnitude greater than the SNPs which have been found in all the human genome projects. And the other thing that you need to think about is the genome of a mosquito is approximately a tenth of the size of the size of a human genome. So what we're seeing is 2 orders of magnitude greater diversity than any of the other major genome projects. So fantastic. Lots of diversity. What does that mean in a public health context? Well, it obviously means that there's incredible standing genetic variation. So we can think of that meaning that any, for example, insecticide resistance variant which is likely to merge is already extant in the population. So any variant which could cause resistance, a simple polymorphic, sort of a simple DNA-based polymorphism which could cause resistance is probably already out there somewhere. Not all necessary combinations will be in the same population, but there's a wealth of data, and so you can see immediately how rapidly these can respond to insecticidal or other controlled pressure. And we can think about how that variation might affect some of these new genome editing approaches. So I'm sure you've probably all been aware of the emphasis of late has been focused upon CRISPR-Cas9 technologies. So essentially the Cas9 is a nuclease. It complexes with a guide RNA, which is shown here. That guide RNA is complementary to an area in your organism of interest genome. And this Cas9 and guide RNA binds to the genome and causes a double strand break, and therefore obviously alters the function or deletes the function of that gene. It prevents transcription. And this is being a favored way of late of thinking about modifying vector populations, so identifying maybe appropriate effector molecules, for example, something that might be a gene which might be associated with midgut invasion by the parasite in the vector or invasion into the salivary glands. Large amount of work is going on to identifying effector molecules which may be amenable to this genome editing approach. But what we're showing is that these guide RNAs are 21 bases long, and they're very susceptible to variations in the complementary sequence. And so what we can do is use the 765 specimens and screen the approximately 13,000 genes we know of in the Gambi genome, and we can identify that there are probably 11,000 genes which have viable CRISPR-Cas targets, so things that could be targeted by this genome editing technology. If we then, if we then account for the variation we're observing, we actually cut that number in half, and that only about 5,000 of those genes have a viable target which doesn't show any variation within the potential CRISPR-Cas binding site. But this is obviously a gross underestimate. We've only looked at less than 1,000 mosquitoes. Very conservative, so we would expect once we sequence more that this number will fall appreciably. But we can think about this in the converse way. Can we identify genes which actually have multiple CRISPR-Cas sites within them, which seem to be conserved, and therefore we can actually try and overcome this innate variant problem by designing multiple CRISPR-Cas sites per individual. So this is sort of taking the converse. We're not thinking about what's the effector molecule. We're actually thinking about which genes it is possible to detect. And what's quite exciting is that we can actually do that, and there are some. So, for example, there's sort of 9 putative sterility-associated genes which seem to have multiple CRISPR-Cas binding sites. So we can actually think about maybe designing genome technologies editing technologies against those. So population suppression, can we use sort of genomic technologies to monitor vector control? I guess the eagle-eyed amongst you will have noticed that's not a mosquito in the top right. What I'm trying to do is take an example from Steve Torr's work and some data from an MSc student, Alan Mahesi, who's actually used more simple genetic technologies to look at intervention impacts in tsetse fly populations, and hopefully then segue seamlessly back into mosquitoes. So this is a study being conducted in Western Uganda, where Steve's team have designed and deployed these tsetse fly traps, which attract the flies in and then kill them through insecticidal action. And they're targeting a riverine tsetse species, so it lives, as the name implies, along the river. And he has areas, or the team have areas, where they implement control with these interventions, and downstream areas with lower mosquito tsetse numbers, where it's not deemed necessary to have this control. And what they find is this is a very, very effective control technique. And so if you look at trap numbers in intervention areas, you get a very rapid tail off in fly numbers in your traps. And the flies can be invisible to your trapping technologies, where in non-intervention areas, you still see appreciable numbers. And what Alan Mahesi was interested in doing is can we actually track genetically whether we can see whether this is truly evidence of population crashes, or potentially are populations remaining at the same effective population size, the same levels of diversity, but essentially become invisible to our trapping technologies. So an analogy with malaria epidemiology would be, have insecticide-treated bed nets knocked populations on the head, or have we induced them to change behavior, and now are we seeing much more biting outside? And so what Alan was doing was looking at molecular markers of diversity in pairs, temporal pairs, either pre and post intervention in this upstream intervention region, and time match pairs from a time match pair from a downstream non-intervention region. And really what you're able to see in the non-intervention region, there's no effective change in this estimate of diversity, but you see this big fall in diversity in the intervention area post-intervention. And so what we're interested in seeing in these very, very diverse vector populations, so Anopheles gambiae populations, is it possible to see similar changes in diversity? And so there are numerous examples from sub-Saharan Africa of decreases in various estimates of malaria transmission and prevalence. And I'll talk briefly about Kenya. And these are data from Western Kenya. No, they're not. They're data from Eastern Kenya, from Kilifi. And this graph is showing pediatric admissions in Kilifi. And you can see they're tailing off between the period 98 to 2008. And that's in some way associated with ITN scale-up. That's also what's illustrated here, increasing ITN, insecticide-treated net coverage in this area, but also the advent of long-lasting nets and ACT therapy. So there are evidence of these falling malaria indices, coincident, at least to some extent, with scale-up of interventions. And at least in this setting, there's some evidence that vector densities have fallen as well. Can we see any trace of that in the genomes? And the answer is yes. We can see really striking estimates of population size crashing in these populations. So what we're looking at here are these are individuals, and we're scanning across one of their chromosomes. This is the left arm of chromosome 2. And this group of individuals all come from Angola, and they're Anopheles kalitzii, and this group of individuals all come from Kenya, from the same sites where we've seen these population crashes and we've seen lower vector densities. And what we're looking at is areas of the genome where these things are homozygous, and that's marked in red, or areas where they're heterozygous, and that's marked in white. And I think you can tell quite readily that what you're seeing here is much higher rates of homozygosity in the Kenyan populations than you're seeing in the Angolan populations. And this homozygosity is a characteristic effect of very, very low population size. So population size is falling. The likelihood of with mating between siblings increases. And so what we're probably seeing here is really close sibling mating in these individuals here. But in general, what we're seeing is a much reduced effective population size, which seems to be coincident with ITN. We can't say it's causal, but at least it raises a possibility if we can embed genomic technologies into monitoring strategies, that we can start to use these things for looking at changes in effective population size or efficacy of interventions in real time. So on to resistance. So what we're showing here are two schematics of mosquito populations. We're looking at a string of DNA here, and each of these beads, if you will, is a polymorphism in the genome of this individual mosquito. So obviously we're very keen to perturb systems. So in we come and we spray insecticides on the walls. And in one of these populations, a resistant allele emerges. With continued insecticidal use, what you may see is that this resistant associated variant sweeps through the population. And what it'll happen, it'll drag behind all the variants which were associated. Because this is happening over a very short time scale, there's not a chance for recombination. So all individuals which have this resistant allele will also share a large chunk of the genome as well. So it sweeps to fixation in this population, and it also hops over the border and starts to increase in frequency in this neighboring population. But it may not be as simple as that. You might get independent evolution of insecticide resistance. So you might have the same variant, associated resistance in two populations, but you can see here it's on a different haplotypic background. So you can have multiple origins of resistance. And there could be various combinations of this. So you could have different origins of resistance with some migration. You could have novel variants occurring outside your area, and you can start to see resistance accumulations on top of the back resistance. So these could be additive resistance variants. They somehow ameliorate or they can somehow ameliorate the fitness effects of the resistance variant. So that's really one of the questions we're asking. Can we identify these high-frequency haplotypes in the genome sequences? Can we associate them, see how often frequently they occur? Can we predict whether they're likely to be found in more than one population? And hopefully then, can we then drive this forward into diagnostics? And this is essentially an analysis which summarizes that search for these haplotypes associated with resistance. Again, we're looking across all the populations from Angola up here through Cameroon and down into Uganda. And we're scanning across the genome and trying to identify areas of the genome which have a signal of big haplotypes at high frequency, a sort of characteristic signal of this selective sweep. And we can see that some selective sweeps seem to be relatively cosmopolitan. They seem to be found in a large number of populations. And this large one here is the KDR mutation in the voltage-gated sodium channel, which seems to be quite common. We see some here, which is just found in Burkina Faso, which seems to be centered on a refractoriness, a parasite refractoriness locus. But we can see others which are clustered around families of glutathionase transferases, quite commonly, and yet more which seem to be occurring around clusters of cytochrome 6 P450s. So we have a range of these signals, and what we've been doing is digging down into these signals and using the biobanks of resistance phenotype specimens we have here to try and validate their association with insecticide resistance. And the first step to do this is to look at those swept regions and pull out the individual haplotypes which are lying underneath them. And so we take a smaller substrate section of the genome, and we can construct these haplotype trees. So essentially we've taken 765 individuals, they're diploid, so we split their genomes into individual haplotypes, and we create these very simple distance trees here. So this is SNP distance between individuals. And so what you'll be able to see, there are some individuals which share essentially the same haplotype here. And these are also sharing very, very close related haplotypes here. So we can identify haplotypes which are very, very common. And what we can see is also on this ribbon here shows how they're distributed across these populations from Angola, Cameroon, and so on. So we can see that this selective sweep is confined to Uganda, this light green here, And what we're showing down here are the non-synonymous changes which seem to be underpinning this haplotype. And so this is the voltage-gated sodium channel. And what we'll talk about now is a little bit detailed of how many origins of resistance and what we're seeing here. So we look in the voltage-gated sodium channel, the target of DDT and pyrethroids, where we have this common KDR mechanism. What we can see are approximately 10 independent selective sweep events. So we see 10 different origins of resistance in these populations. Some of them are very soft selective sweeps in that they're locale specific. So this is just found in Uganda. And you can see here some are much more cosmopolitan. They're found much more widely. And that's illustrated in this map up here. So we can see one haplotype here, F1, which is essentially found all the way from Guinea through Burkina Faso, Cameroon, and down into Angola. So one gene which has occurred, one mutation which has occurred on a haplotypic background and spread really throughout West and Central Africa. We can see another example here of one which has spread between Uganda and Kenya, or vice versa. And remember what we were saying here, there was a big barrier to gene flow here. This is the East African Rift Valley, which goes here. We don't find populations of Gambi, but we are able to see resistance mutations hopping across areas which are inimical to mosquito breeding. So we can track multiple origins and their spread. But again, this is a gene we've known a lot about for quite a while. What's the novel insight here? Well, the novel insight, I think, comes from when we look at the detail of this, is that we look at, say, for example, this haplotype. This is the 1014F, what was formerly called the West African KDR mutation, one that's very, very widely spread. What we can actually start to see are other haplotypes, if you will, which are embedded within that, which are indicative of an overlaid selective sweep. And so if we look at the variants here, we don't see any synonymous variants. They're all non-synonymous. And some of them have reached quite high frequency and are quite widely distributed. And so this mutation here is in codon 1575, and this essentially doubles the effect of KDR. So it increases the likelihood of surviving a DDT or a pyrethroid exposure twofold. And what you can see here, there's another series of really quite high frequency and quite widely distributed mutations which are lying along this background. And a number of these, through Eric and Ian's work, are actually validating their association with the resistance. So even in this scan of non-phenotyped mosquitoes, we've already got probably 3 to 4 new resistance-associated variants or likely resistance-associated variants in KDR. So from having one SNP mutation in the last 15 years, within sort of 6 months of this work starting, really digging down into this, we've got 3 to 4 new ones just within what we thought was a very well-characterized locus. And that's something which probably could have been done with comparative genomic approaches or just sort of targeted sequencing. But we're also revealing quite a lot, I think, about sort of some of the other resistance variants. So another selective sweep was observed around GST cluster. And what we see there are essentially 4 independent selective sweeps. This one here is cosmopolitan. You can see it's spread all the way from West to East Africa. What we think is the mutation underpinning this selective sweep is a polymorphism in the gene within that cluster of GSTE2. It's a mutation in codon 114, which seems to confer higher DDTase activity than the 2 wild-type forms. So this is probably driven by DDT exposure. We have a Ugandan-specific sweep, with which we have diagnostic markers now, and this seems to be driven by expression level variation in GSTE4. And there's another sweep here. It's another mutation in GSTE2. It's a Valian change at codon 119, which confers, which Eric Lucas's work has just showed, seems strongly resistance associated in both Burkina Faso and DRC, this area where we have very, very poor sampling. So we're already generating resistance-associated variants, and we're showing in certain genes, we're seeing evidence of relatively small number of origins and continental spread. And what we now have, we have sort of high-plex assays of 2025 SNPs, which we're able to push through and get sort of much more detailed resistance genotyping information. But to go back to the cytochrome P450s, which I said we had seem to be consistently upregulated in principally pyrethroid-resistant phenotyped mosquitoes or populations. What's the pattern there? And I guess before we get on to this ribbon diagram, I think what's probably most illustrative is this picture up here. And what you're seeing, you're failing to see those interconnections between populations. So what we're seeing is these 8 selective sweeps we've identified are all population specific. So this is a Ugandan specific one. This is essentially Burkina Faso specific. This is a Cameroon specific one. We're not seeing evidence of cytochrome CYP6 resistance mechanisms evolving, emerging, and spreading. It's a much more localized story. So it's a combination of small number of origins of certain resistance mechanisms and global spread. But in this case, for the CYP6s, it's much more localized emergences, and they don't seem to spread so well. So it's, again, this idea of selecting from standing genetic variation. And this probably underpins why we are, in the initial GWAS studies, had very poor resolution of actually trying to identify CYP6 associated variants. But we've got to a point now, I think, where we have a suite of markers, and within the next 6 months we'll have, let's guesstimate, sort of probably 25 resistance-associated variants, that we can actually start to develop these risk maps. And that's the next stage of this work, to actually sort of develop risk maps, genotype-based risk maps, and some associated estimate of error for use in predictive. And I'll talk very briefly about some of the teams who work in sub-Saharan Africa to develop these technologies. So in the last sort of 5 minutes or so, I'd like to maybe sort of pitch where we think this is going and how our more public health-oriented work can really drive some of the questions. And when I introduced this slide earlier, I said that we sampled widely across sub-Saharan Africa. That was a little bit disingenuous because we didn't sample positively or proactively across sub-Saharan Africa. We sampled, our sampling unit was the freezer, and we went to various, principally research in the Northern Hemisphere, and asked them if they had any specimens hanging around in their freezers which we could use. And I think there's a growing awareness that that's really is a suboptimal sampling strategy. As we're moving into sort of from population genomics is becoming more common, I think there's a growing realization that a genome is really only as good as its associated metadata. And to illustrate this is a really nice study from Nick Thompson from the Sanger group. And essentially what they've done is they've taken a Shigella bacteria from the National Type Collection, and they've done a genome sequence, which in itself is pretty much a trivial exercise. It's these bacterial genomes are relatively simple to sequence. But what actually means this is why this is a Lancet paper rather than anything else is that it's actually the associated metadata. And it's published in 2014, obviously the centenary of the outbreak of the First World War. And the story really is that this submission to the National Type Collection was from a soldier who died of dysentery in the First World War. So it really sort of, it tells the story of the war. And so I think that's really what we need to think about is how we move forward. What is the associated metadata? In our case, what's the epidemiological setting we're sampling from? Do our individual mosquitoes we're submitting have infection status, resistance status? Do we have any behavioral? Do we have any collection data from them? And I think that's really where our skills lie in constructing the hypothesis around that metadata. And just to give a sort of a couple of illustrations of how this can evolve. The one collection of the 1,000 genomes which didn't come from a freezer, which was collected by Henry Mwedji and Craig Wilding from the group in Nagongera in eastern Uganda. This is an area of Africa which has an incredibly high annual entomological inoculation rate. So we estimated that the EIR in this population for P. falciparum was 125 infectious bites per person per year. And that is, for a number of years, been working with teams from Makerere and UCSF, and they've been characterizing infection in children, in a cohort of children in this area of Tororo district. And what they're observing that certain clusters of children, and each of these points is a cluster household, a cohort child household, that certain children in this area might get 12 malaria-attributed fevers a year, and that others might be getting 2 to 3. And so we were actually able to sample across these hotspots and cold spots of transmission as well before the submission sampling. And that sort of stuff we can move forward with it in due course. And so Nagangara is also very interesting because it's been an area where we've done resistance work, and we see extensive pyrethroid and DDT resistance in the primary vector there, Anopheles gambiae. And it's also an area where we know a lot about the interventions which are going on. So this is a series of 3 different malaria metric indices collected from October 2011 to 2016. So we have in green an estimate of the daily human biting rate. In blue, we have incidence in those cohort of children, and we have in red the test positivity rate at the local clinic. So we have very good time series of malaria metric indicators, and we know a lot about the interventions that go into these populations. So we can see that in this area here, in yellow, there was an LLIN distribution, and you can see very little evidence of any perturbation on these malaria metric indices, possibly driven by pyrethroid resistance here, or impacted by pyrethroid resistance. But when we see bendiacarb spraying going on in subsequent years, we can see this big tail off in all the malaria metric indices. And we can build our genomic sampling into this framework, and that's what we're doing in a couple of studies moving forward. So for example, this is a figure from a patch very similar to the other one. In this case, this is just test positivity rates. What we see here in yellow are a series of bendiacarb spray rounds. The National Malaria Control Program in Uganda had to withdraw spraying from this area around a patch, and before they did that, they had a large ITN coverage rollout campaign, and what you can see is that that didn't actually maintain malaria metric indices at low levels. You saw this massive rebound. And so what we're doing in Uganda, so this is a study with Janet and Hilary and Sarah Stedeker from London School, we're looking at randomized control trials of new interventions, new insecticide-treated nets which are treated with PBO. And in this area here, which is one of the former spray districts, we're also looking at a randomized control with Sarah Stedeker of what happens when you withdraw IRS interventions. Can you use behavioral support therapy? Can you use mass drug administration? So we can embed uniquely, I think, our control programs and our genomic programs into active interventions, and we can actually see, well, can we see the advent of resistance? Can we see evidence of changes in effective population size in real time for using as predictive outcome measures? And I think that's just taking Uganda an example, but I think, again, because of the breadth of reach of the school, this is something we can conduct much more broadly. So with NIH and a GCRF award, we're working with groups in West Africa and East Africa who are actually doing routine sampling. So, for example, Edie Constant is involved in projects testing new active ingredients from Sumitomo. Erica Chomo is based in Western Kenya, working with Faker Takal on various projects associated with indoor residual spraying and long-lasting nets. So we're working with actors who are actively engaged in implementation research, and they are able to develop these time series. So they'll have surveillance sites which are sampled over very, very many years, and with sequencing through the sort of follow-on project called the Vector Observatory at the Sanger Institute, what we'll be able to do is both respond very rapidly to when resistance emerges, because we'll be able to sequence and identify resistance-associated variants in real time and develop understanding of mechanisms, and obviously push these into surveillance with these folks. But we can also do something which is more proactive, I hope, in that we can actually start to understand how do mutations evolve. We've shown you how we can track them, whether they evolve singly or multiple occasions. What's the underlying evolutionary process which drives that emergence? And therefore, actually, can we actually think about ways of mitigating resistance before it emerges? So thinking of how we might want to implement strategies and use the genomics to inform that. So that's really a bit of a prospect for, but I think that's the direction of travel. I think once we are embedded within these interventions, I think there's a real possibility for really harnessing the power of genomics. So just to conclude, I think we've shown that this high diversity, this surprisingly high diversity in Anopheles gambiae populations has really strong implications for the emergence of insecticide resistance multiple occasions, and also for transgenic genome editing based approaches. I think there's potential for using genomics in real time to detect small changes in effective population size, not necessarily the huge population crashes we've seen in Kalifi, but I think we'll have the resolution to use it in real time to monitor changes over time in a predictive fashion. And we've seen this sort of this hodgepodge, this rattlebag of resistance emerging both on a singly and spreading widely, or large numbers of sort of local emergences of resistance. And I think our next challenge, and I think one which we are, as a school, are very well placed to do, is to actually integrate these interventions integrate our genomic studies into public health interventions. So thank you for your attention, and also just to thank sort of many of the key people who work. So a lot of this work, I should say, is done in associating David Wheatman, so big thanks to him, and also groups across the school, in East and West Africa, and at the Oxford Sanger group. So thanks to all those, and thanks to you. Thank you.
With your difference between your P450s and your KDR, with the difference between P450 and KDR in terms of numbers of origins and spread, is that a function of time since the resistance was selected, or do you think there's something different going on there?
I think it may be a function of diversity, actually. So the voltage-gated sodium channel where KDR emerges is in the central mirror. It's relatively low diversity. The P450s, if we sequence through those are really at the upper end of the variance. So they'll probably have a SNP every base. So there's a huge amount of standing genetic variation in P450s. It's much more restricted in the voltage-gated sodium channel, maybe through selective constraints on because it's a neurotransmitter. So I think that's probably what may be driving it, that sort of marked difference in variance. So there's this wellspring ready to spring up as soon as you impose a little bit of selection on the P450s. Mark?
I'll just shout if that's all right.
You won't get onto the live stream.
Oh, right, okay. That's on it. Okay, I won't shout. I'll talk slowly. There's new insecticides coming on the market, and they're going into a sort of a naive population, if you like. How does this type of information, or how can this support the new insecticides in sort of preventing resistance, if you like?
Well, I think there's obviously one way, and obviously very much the work you're doing. We can identify resistance variants here, which seem to be showing signatures of selection very rapidly, and you can also get them into your validation approaches. But what we're also doing is we're actually already embedded within some of the new trials. So I mentioned Edie Constant, he's looking at SumoShield, and so we're actually embedded within the sort of the early stage trials. And so we can see these emergent selective sweep signals within sort of real time, if you will, over the course of a very much an experimental trial, I would hope. So we can hopefully get ahead of that before it reaches market. We'll have an understanding of what's the evolutionary process which is likely to lead to resistance.
You intimated that one of the reasons you see insecticide resistance is the huge diversity in your populations, but I wonder whether that's just a function of the sampling of a very large population size. And if you overlay that with the local emergence of resistance, is there not the worry that actually some of the drugs, insecticides that are being selected, are actually quite easy to be overcome? And isn't this an issue that we've also seen in drugs as well, that some of the mitochondrial drugs are also easy to overcome? And therefore, should we be trying to screen some of the drugs for actually not being able to be overcome by using the genetics of when they're implemented?
Yeah. Yes, so the effective population size is very large in these things, and that's obviously reflected in the genome diversity. And I think it's true that standing genetic variation does mean that for many of these active ingredients, use as a monotherapy may well very rapidly select for resistance. And I guess that's been the repetitive experiment we've done over time using insecticides as a monotherapy. We very rapidly see the emergence of resistance. So yes, I think what we need to be is much more creative about the use of PBO synergists, about sort of mosaics and combination therapies. Take a lead from the ACT malaria book, really.
Yep. Good afternoon, Professor. Thanks for the presentation. My name is Aboa John, and I'm from Uganda. I just wanted to pick your thoughts on what you think could be causing this development of resistance in these mosquitoes. Could it be the way we are applying the insecticide use? We have different strategies we are using there. Could it be the way we are applying the intervention of using insecticide use, or could it be maybe the problem with the manufacturers in terms of dose of the pyrethrum they use to spray, something like that? Thank you.
I guess the simpler answer to that is probably all of the above. What is particularly important about Uganda is where we mainly work in eastern Uganda, and that is also a major cotton growing area as well. So I think that's another actor we don't really have a good handle on whether the selective pressure is being driven by the public health use, or is it also being contributed by agricultural use as well. And certainly in cotton growing areas, there does seem to be this emergent association between emergent resistance and insecticide use on cotton. So I think we have a very poor understanding of what's driving emergent resistance. I think some of the trials we're doing, where we're looking at differential insecticides across relatively small areas, we might start to untangle some of that, and whether we can see whether it's a public health driver or it is a more an agricultural driver.
Hi, is that on? You showed some really nice data of your SNP analysis, and I was just wondering, I think you might have touched on this in the last slide of your talk. Have you got any more evidence for gross genomic changes in your isolates, whether it be indels or intramolecular recombination?
Gross stuff we haven't really dug down into. We're just starting, or Eric Luce, to be fair, is just starting to look at copy number variants in some of these things. We do see, if we do genome scans, we do see lots of sort of CNV variation across these, and some of them seem to be actually within gene families which we know to be resistance associated. But thus far, we haven't really done anything about sort of genome structure or genome architecture. We have a small handle on copy number variants, but that's about it, I'm afraid. It's been very much SNP-based at the present time. But obviously, it's the next step.
So, malaria and anopheles have very much led the way amongst vector-borne diseases in terms of these kind of big-scale genomic projects. Do you think that this could serve as a template that could, or maybe actually should it be, repeated for other vector-borne disease systems?
Yeah, I think so. I don't see why not. And it's our intention, obviously what we're talking about here is very much Anopheles gambiae-centric. In the next phase of the sort of the Sanger renewal, there's an opportunity to sequence somewhere between 30 and 300,000 mosquitoes. We're not quite sure the price point at the present time. And the intention there is to very much move into sort of Southeast Asian vectors, and that's obviously the next step. And I guess we'll see very, very similar patterns of diversity in there, and hopefully generate some of the same tools. With other vector species, I think the power would also be there. Whether there's the public health driver, certainly I think in AD systems where we have started to get a handle of sort of cosmopolitan spread and emergent resistance, I think that would be the obvious next step. Some of the sort of, I'm not quite sure they call them niche vectors, tsetse, black flies, whether there's the public health demand for it there, I'm not sure at the present time. But certainly in the AD vectors and Culex vectors of LF, it would be an obvious next step.
Thanks, Martin, for your talk. Am I the last question? The last question. This is, I hope, something that I just didn't miss listening to you. When you looked at the 1,000 Genome Project, did you notice if the mosquitoes sorted out between those that were caught outside and those that were caught inside when you did comparisons?
We didn't explicitly have that data here. In phase 2, we have one population which was collected in the Gambia, and some of those were collected inside and some were outside. I think the problem with that phenotype is inherently messy. I think you can sit outside a house, you can intercept a mosquito on its way into the house. It doesn't necessarily mean it's an outdoor biting population. And so I think that the collection from the Gambia, the collection was done only 2 to 3 meters away from a house, and so I'm not convinced that's a very strong phenotype as such. I think what we need to see is reproducible phenotypes. Can we do, say, mark release recapture and generate phenotypes which are consistent in one group and the other before we can really start to do sort of GWAS sampling approaches for behavior. It's very it's not intractable, completely intractable, but it's a problem I wouldn't want to delve into just yet. Okay. Well, thank you very much. Thank you.